Hey guys, welcome back to our channel where we bring to you the science behind the COVID pandemic that we are currently facing. If no, you haven't seen the first episode of this series, please go have a look. If yes, then let us learn about SARS-CoV-2 in detail. So in this episode, I and my colleagues Disha, Sakshi and Purvesha will be talking about the structure, the source and the life cycle of SARS-CoV-2 in detail and also the organs that it affects.
Let's talk about the source of coronavirus. Exactly what is the probable source of SARS-CoV-2? In December 2019, a pneumonia outbreak was reported in Wuhan, China, which was traced to be a novel strain of coronavirus and was named as 2019 nCoV by the World Health Organization, further renamed as SARS-CoV-2. This virus has a 96% of similarity to a bat coronavirus and hence it is also suspected to be originated from bats as well. However, in recent reports, China claims that the seafood markets in Wuhan are not the only source of this virus. And they said, but to be honest, we do not know the source of this virus now. A phylogenetic study published in 2020 indicates that pangolins are a reservoir host of SARS-CoV-2 like coronaviruses. However, there are no such evidences that link pangolins, intermediate host of SARS-CoV-2 like coronaviruses. While there is scientific consensus that bats are the ultimate source of coronaviruses at this moment, it is hypothesized that SARS-CoV-2 like coronaviruses originated in pangolins, jumped to bats, jumped to humans, resulting in the coronavirus.
Let's talk about the structure. As discussed in the previous episode, coronavirus virions consist of 4 different types of proteins: the S protein, the M protein, E protein and the nucleocapsid protein. The S protein here produces a trimeric structure which is fusion protein and it mediates the attachment to the host receptor ACE2 shown in the figure. The M protein is the most abundant structural protein. It is a small protein and which has 3 transmembrane domains. Recent studies showed us that the M protein exists as a dimer within the virion and it promotes the membrane curvature. The E protein is found in very small quantities within the virion and it facilitates the assembly and the release of the virus. The N protein constitutes of the only protein present in the nucleocapsid. It is composed of 2 separate domains, the NTD, the N-terminal domain and the CTD, the C-terminal domain, both capable of binding RNA.
Now, let's have a look at the life cycle of the coronavirus. The attachment of the virion to the host species is determined by the interaction of the S protein and its receptor. The S protein receptor interaction is primary determinant for the coronavirus to infect a host species and also determines the tissue tropism of the virus. The receptor for SARS-CoV-2 is ACE2, which is angiotensin converting enzyme 2 on the epithelial cells. In this diagram, the numbers indicates the steps in the life cycle of coronavirus. After the receptor binding in step 1, the virus gains access to the host cell cytosol through acid-dependent proteolytic cleavage of the S protein. In step 2, there is fusion of the viral and cellular membranes. In the next step, that is step 3, the viral RNA is directly used for translation of the replicase gene, also known as RNA-dependent RNA polymerase (RDRP). Then many of the non-structural proteins assemble into replicase transcriptase complex to support RNA synthesis and ultimately are responsible for replication of the RNA and transcription of subgenomic RNAs as shown in the step 4 and 5. Following replication and subgenomic RNA synthesis, the viral structural proteins S, E and M are translated and inserted into the endoplasmic reticulum indicated in the step 6. These proteins move along the secretory pathway into endoplasmic reticulum Golgi complex. Then the viral genome are encapsulated by N protein and they bud into the membrane of the Golgi complex containing viral structural protein indicated in the step 7. Mature virions are formed after the assembly as in step 8 and then are transported to the cell surface in vesicles and released by exocytosis. In this way, the virus multiplies inside the host cell and is released by exocytosis to cause further infection.
What are the organs that SARS-CoV can infect? In a study, the scientists looked for the SARS-CoV in tissues of the patients that died of SARS. Results suggest that, in addition to the respiratory tract, the gastrointestinal tract and the other organs like kidneys and sweat glands, which are detectable of SARS-CoV, can also be possible targets of SARS-CoV-2 infection, and thereby it can lead to the virus transmission that may be excreted via feces, urine, and sweat. The complications caused by the above-mentioned organs can be the severe lung condition causing the low amount of oxygen to go to your organs, also known as acute respiratory distress syndrome, the organ failure in many organs, blood clots, and also acute kidney injuries, etc.
So welcome to Rumor Humor, where we bust rumors about COVID-19 that have created unnecessary panic. There have been speculations, thanks to such fantastic memes, that the virus was developed in laboratory and released either accidentally or as a form of bioterrorism. Recent studies have however shown that though SARS-CoV-2 virus may bind to the human ACE2 receptor of lung cells with strong affinity, the interaction is not ideal. The protein sequence of the receptor binding domain of SARS-CoV is not optimal for perfect receptor binding. Thus, the high affinity binding of SARS-CoV-2 spike protein to human ACE2 receptor is most likely the result of natural selection on a human or a human-like ACE2 receptor that permits optimal binding, which is not perfect. This is a strong evidence that SARS-CoV-2 is not the production of purposeful manipulation. Additionally, the genetic data irrefutably shows that SARS-CoV-2 is not derived from any previously derived virus backbone. This theory is also supported by the Directorate of National Intelligence, United States of America. So that's the rumor busted for today.
Quiz questions for this episode are: what are the function of S protein? Also, answers to the previous question in the last video are in the description box below. Please check.
This brings us to the end of today's episode. The links of the information that we have used in this episode is given in the description box below. We were really overwhelmed by the response that we received for the first episode. Thanks a lot for your support. Please do like, subscribe and share the video with your family and friends. However, this does not end over here. We will soon be back with a brand new episode. Till then, take care, stay safe and stay tuned. Bye bye.